Tigerson here, and recently we have received many reports on mitosis and meiosis in the area. We now go to Specialist Nathan Larson with the report.
Thank you, Tigerson. Well, you see, there are two different types of cell division. The first one we're going to focus on is mitosis. We have created some diagrams for the viewer's help. Thank you. So this is a cell. This is the cell membrane, and this is the chromosome inside of it. So the first step of mitosis is interphase, where the cell grows in size. It's not hurt. No, it's not. It grows in size, and it duplicates its chromosomes to become, and these are just floating around. So the first step of mitosis is prophase, where all of the chromosomes line up with their matching pair and become sister chromatids. So then we go to the next step. Is called metaphase. So in metaphase, all the chromosomes line up along the equator, and they're held there by these two centrioles. So now the fun begins. In the next phase, anaphase, the centrioles have spindles attaching to each of the chromatids, and they separate them. They pull the chromatids apart, each into their own half. And now imagine this with all these chromatids times 4. With 44 or more of these. That's how the human cell goes. Now for the last stage of mitosis. It is called telophase. So the cell membrane closes, the cells separates. Just like this. Now we begin interphase all over again. But that is not the only type of cell reproduction. Now we will present meiosis, a different take on cell reproduction. So, as you can see, we've decided to upgrade our cell model, and now we will begin meiosis. So, the different thing about meiosis is that instead of starting with just two chromatids, or chromosomes, we now start with 4. One of each is from the mother and from the father. And likewise, mother, father. So these two are connected. And then in prophase 1, they swap different alleles as such. So they just switch out different alleles and crossing over. So now there's more different combinations in the genes. So now we will begin metaphase. But remember, this is the first metaphase. So, like in mitosis, they line up along the equator with two centrioles lined on the outside. In anaphase 1, like in anaphase mitosis, they split apart. But the difference here is that instead of ending one sister chromatid in each different cell, there are now the homologous chromosomes split up into 2 sister chromatids in each cell, but there's still 2 cells. Now, we'll begin meiosis 2. So, it is very similar to meiosis 1. And the centrioles line up, and then metaphase 2, the chromosomes line up, and there go the alleles. I'll put those back on. No, I won't. Yes, I will. the chromosome line up along the equator, and again in anaphase 2 they split apart. And telophase 2 creates 4 different cells. So now here's the tricky part. The difference between mitosis and meiosis. In meiosis, we end with 4 cells, not 2. And imagine each of these cells having each 23 chromosomes. All one from a parent, one from a father, and one from a mother. Imagine all the endless possibilities we could have with those combinations and with the crossings over in the first prophase. We'll now go back to tiger sin.